Okay, so Friday afternoon, let's get started, shall we? We were almost done topic 8 last day. There's a few more things to kind of go over, more or less regarding ATP. And I know we've mentioned ATP a few times this semester, and you probably know a little bit about it from high school, but I want to go over a little bit more information regarding ATP. So just a quick recap of some things that we talked about on Wednesday. First, we talked about catabolic and anabolic reactions, also sometimes referred to as endergonic or exergonic and endergonic reactions. And one of the concepts we talked about was this thing here, delta G. So delta G means the change in free energy. And this is the amount of useful energy that is either released or absorbed in a chemical reaction. So there's more energy than that kept in chemical bonds, but this is the amount that can be used for work. And in particular, in this class, we're thinking more about cellular work, and I'll talk about cellular work here in a few minutes. So exergonic reaction on the left, energy is released. We're starting at our reactants have high energy and they lose energy, and so the value is negative. Endergonic on the right, the reactants are at a low energy level. They absorb energy, or the reaction requires energy before it can actually proceed, and therefore the delta G is a positive number. So I know I had also talked about this slide here, and as a way to think about a cell, we have nutrients and energy going in. The cell is building itself to be an organized living organism, and waste products and heat are being sent out. And I'm going to skip this. You can take a look in the textbook. It's talking about cells and equilibrium and all that. It's kind of not so relevant to what I'm talking about here. And this is where I finished off, and I want to talk a little bit more, like I said, about ATP and a little bit about cellular work. Okay, so here's our cell. It's taking nutrients. If you're a human or another heterotroph, usually that means you're eating other organisms for food. So we consume food, and our food has protein and fats and carbohydrates in it and other nutrients, but that's primarily where we get our energy from. If you're a plant, you might get energy from light. If you are a weird organism, some sort of a weird bacterium, you might get it from minerals. But all these things go into the cell, and the cell is going to take these things, and as it is oxidizing the fuels, usually what it does is converts them to a useful cellular form, which is ATP. We're going to talk about ATP. Many of those organic molecules are converted into more organic molecules, so more carbohydrates, or fats, or proteins, or nucleic acids, of course. And that's what the cell is doing. And all of this takes energy. Some of that energy is lost in waste products as chemical bonds, and some is lost as heat to the environment. So bottom line is, as we said last day, if you want to stay alive, if you want to stay organized, you need to continually consume food, and that food is going to be used to give you cellular order and allow you to do cellular work. So let's talk a little bit about ATP and cellular work. So here is ATP. So ATP stands for adenosine. So this here is adenosine right here. Adenosine. So adenosine is that nitrogenous-based adenine attached to that pentose sugar. And triphosphate, so T and P, means we have 3 phosphate groups. So we have 1, 2, and 3. And they're all attached together. So one thing I noticed right away when I look at a molecule like this is I notice it has a whole bunch of oxygens attached to those phosphate groups. And these oxygens, many of them have charges. So this to me is, it means something. You probably know this too. If you have lots of negative charges, due to the laws of chemistry, they're going to be wanting to repel each other. So what does that mean? It means in order to make this molecule, it means we've had to store a lot of energy in these bonds here. So those 2 bonds there are called high energy phosphate bonds. High energy phosphate bonds. And these high energy phosphate bonds can be used by the cell for a whole bunch of things. And we're going to call all of those things broadly, we're going to call them cellular work. And that's kind of the next part of something I want to talk about. But a little bit more about ATP. Often it's abbreviated, sometimes like this, a little cartoony structure. And you can see what they're showing is the ATP is being broken down, and you get inorganic phosphate. So this little character here stands for inorganic phosphate. So inorganic phosphate looks something like this when it's in solution, and it has no carbon in it. That's why it's inorganic. So you put the little I beside it. Sometimes it's just shown as a P with a circle around it. But you can see we broke one of these high energy bonds, and now we have ADP, so adenosine diphosphate, and we've released energy, and that energy can be used to complete cellular work. So a little bit more about that. What do we mean by cellular work? By cellular work, we mean all sorts of things. Often we are talking about moving things. So we've talked about moving things when we talked about membranes. We were talking about transporting ions across membranes, so that's active transport. So up here is active transport. And you can see the example they're showing there is actually the sodium-potassium pump is what they're showing there with that example. You can see that ATP is getting hydrolyzed to its components, ADP and inorganic phosphate. Other cellular work, as an animal, we move around, we have muscles, all of that takes a lot of ATP, and other things can get moved by motor proteins. So you can see in this example down here at the bottom, you have a vesicle that's being moved on a microtubule, and that is cellular work. So usually work in the terms of moving things makes sense to people. But a lot of cellular work actually has to do with something called energy coupling, and that's something I want to talk about a little bit here in a moment. So this diagram is trying to show you what energy coupling looks like. And what I want to do is I'm going to draw something for you on the whiteboard here in a moment. That's an actual example of some energy coupling. So this is showing you have on the left, just focus on the left at the moment, you have exergonic reactions. So you eat a meal, right? So your meal, you have some macromolecules in your meal. So if you eat a donut, it's going to have some carbohydrates and some fat in it as the primary macromolecules, not a lot of protein in a donut. This gets broken down and we make ATP from it, right? And to make ATP, we also require ADP plus inorganic phosphate. So that ATP energy is now going to be used for cellular work. And often that cellular work means that we're coupling the exergonic reactions to drive endergonic reactions. So we're building something with that ATP after we harness the energy from the breaking down reactions. So in this case here, you can see my ATP is going in to these anabolic processes. We're making new macromolecules and doing cellular work. And of course, the ATP gets broken down and we regenerate our ADP and inorganic phosphate. So like I said, I want to show you an example of some cellular work here in a moment. So if you have, you might want a piece of paper because I'm going to draw something here. I'm going to show you how the synthesis of glutamine works. It's an amino acid. So you can see this is kind of just saying, how is the ATP used? The ATP passes off its phosphate groups. It energizes the recipient somehow, and the recipient molecules are more reactive. And this whole process is called phosphorylation because you're putting a phosphate group on something. So let me show you an example. And what we're going to do is we're going to draw this out. So I'm going to pull up my whiteboard here. You can see I have a title. It says energy coupling. Example, the synthesis of glutamine. Okay, so let me show you how we can synthesize glutamine. So what we do to synthesize glutamine is we start off with an amino acid called glutamate. So I'm going to draw that for you right now. And it looks something like this. I won't ask you to ever draw this big chemical structure or anything, but what I want to show you is what's going on with this reaction, because sometimes it's important to kind of understand the details in terms of what is going on. So there's the amino acid. This is glutamic acid. So glutamic acid, that's a U. Glutamic acid. And glutamic acid has an abbreviation. it's GLU. Okay. Now, to make glutamine, we take glutamic acid and we combine it with ammonia. So ammonia looks like this. So there's ammonia. Okay, and so then we make glutamine. Okay, and glutamine looks like this. So it's very similar to glutamate, you'll see. But now we've added that extra amine group, basically, is what we've done. So let me draw this out for you. All right, so that amine group ends up on the bottom, and your glutamine looks something like this. Okay, so this is glutamine. And the abbreviation for glutamine is GLN, by the way. So it looks something like this. This is not a completely balanced reaction. I'm going to balance it for you. The last component of this reaction is water. I don't usually care too much about water. Cells are bathed in water, and it doesn't really matter. It's not something we care about too much. I mean, unless we're dehydrated or something, but a little bit of water in a reaction doesn't usually have anything to do with the energy anymore. So this is your a typical kind of chemical reaction. Now, one thing to know about this reaction is the energy. So let's take a look at the energy. This goes back to these delta G things we were talking about. So this particular reaction, the delta G is plus 3.4 kilocalories per mole. So remember that plus sign, okay? Remember the plus sign we were talking about before, and we said that the reaction, that means that the reaction requires energy to proceed. Reaction requires energy to proceed. Okay, so that's kind of the context here. We have a reaction, we'd love to do it. Sometimes our body needs to synthesize this amino acid. So these are amino acids, by the way. I'll just maybe write that down here. Amino acid and amino acid, just in case you missed that part. So sometimes our body needs to synthesize glutamine, and we don't have energy. So we've got to use energy. And where do we get this energy from? We get this energy from ATP. So let me just move that up a little bit. Just going to shrink it a little bit. And let's talk about ATP. So we'll call this reaction number 1, maybe. And our ATP, when it gets hydrolyzed, so ATP can get hydrolyzed with water. And what do we get when we hydrolyze ATP? We get the components of ATP. ADP, we get inorganic phosphate, and that's what our second reaction looks like. Okay, so what about the energy for reaction number 2? So our delta G for this reaction is actually a negative. It's negative 7.3 kilocalories per mole. So this is great. So remember what that means. It means that energy is released. So that means that's a favorable reaction as well, and it can happen spontaneously. So I'm going to shrink this one more time just so we can fit the last part on here. Well, hopefully everyone can see everything on there. I think that's probably just enough room. So what you can do is you can couple these two reactions. So I'm going to kind of basically add these two things and add these two reactions together. So if you take a look, by the way, if you look at these reactions and if you balance them out, the water basically cancels out. You've got a water on each side. So again, usually I said water doesn't make a big difference. So down here, we'll call this the coupled reaction. Okay, so the coupled reaction, we're just going to add everything up. I'm not going to redraw all the chemical structures, but what I am going to do is use their abbreviation. So we start off with glutamic acid, that's GLU, remember? Then we have ammonia, and we have ATP. And our final products, we get glutamine, GLN, so that's something that we do want. We have ADP, we have our inorganic phosphate, and I guess that's everything, yeah. The last thing to consider is our energy. So overall energy for this reaction, what just happened there? Let me try that G again. Okay, for some reason it wants to turn my thing into a circle. Let me just scratch that out and we'll try that again. Delta G. Sorry about that. Let me try that one more time. Scribble that one out. Delta G. There we go. Delta G. And if you add these two things together, negative 7.3 plus 3.4, what do we get? We get negative 3.9 kilocalories per mole. And that's what our final reaction looks like. So you can see that the final reaction is favorable. So maybe that's my last note to put on here. And we can say that this is a favorable reaction. And maybe you can say that the reaction proceeds and we don't need any extra energy. We've provided enough energy for it. So that is really what energy coupling is all about, is where we take an endergonic reaction and we couple it with an exergonic reaction, so the overall thing is favorable. Just a second, okay? I'm just going to pause this for a second. I'm working at home. My son is looking for something. All right, sorry about that. Yeah, so this is energy coupling. And it's a little bit more complex and maybe not as easy to visualize than actual movement. But this is what most of cellular work looks like, is coupling endergonic and exergonic reactions. Okay, so these concepts, by the way, are going to come back when we get to respiration and photosynthesis, and that's going to be after the reading week. And I'll probably come back and remind you about some of these things a little bit later on. So just keep in mind, like I said, I'm mostly for this topic throwing out some concepts that are going to come back and be used for respiration and photosynthesis. So that's energy coupling. And that's actually the end of topic 8. So that's where I wanted to be last day, but I guess we just got a little bit behind and that's okay. And topic 9 is short and we should be done in plenty of time to get off early for the reading week. Topic 9 is on enzymes and it's actually the same chapter in the textbook, by the way. And I usually separate them out because with enzymes, we are talking about one specific thing that's worth it to kind of focus on. And usually it takes a whole lecture to get there. So let's talk about enzymes. So enzymes are important in your cells. And you probably know that your cells have hundreds of reactions and each of those reactions are facilitated by enzymes. So let's talk about a little bit what enzymes are. Enzymes are catalysts. And what does that mean? It means they speed up chemical reactions. So let me show you a couple of chemical reactions that are actually normally very slow, and I'll show you what enzymes do. So here's penicillin. Penicillin is a drug, and it does degrade. It does degrade slowly. Its half-life in an aqueous solution is about 84 hours. But if you think about it, the average bacterial cell only lives and divides maybe every 20 to 30 minutes. So normally this drug is stable enough and it's not a big deal that this happens. Some bacteria, unfortunately, have resistance and they have enzymes that will break down the penicillin. And these are actually some of the fastest enzymes ever recorded. Some of them apparently degrade 1 million molecules per second. And so now you have organisms that are resistant. They can degrade this drug very quickly, and the drug becomes non-functional and useless. So kind of scary, really. And it's something we certainly think a lot about in the medical community in terms of what kind of resistance organisms have. I'll show you another example. Here's an example from the textbook. This is sucrose. So we were talking about sucrose a few weeks ago as being a disaccharide. This is basically table sugar. And sucrose is made out of 2 monosaccharides. And so if you put sucrose in an aqueous solution, if it's sterile, it would actually take years to break down. You put it in your body, and we have an enzyme called sucrase that will break it down in milliseconds. And so you can release the subunits. The subunits are glucose and fructose. So it makes sense. We don't have years to digest our meals. Normally, we want to have that energy and nutrients today. So it's important that we have enzymes. Enzymes speed up reactions. So I want to get to this other part here about lowering energy barriers in a couple of minutes, but a few other things to say about enzymes. And just a reminder, right? You can see this gives us a delta G with this reaction. So this reaction is favorable. Even if it's slow, it doesn't, slow speed has nothing to do with energy in a reaction, by the way. So enzymes are catalysts. It says here they lower the amount of activation energy. We'll get onto that in a minute. But a couple of things to point out about enzymes. So let's talk about this activation energy thing first, okay? So what I want you to do is just focus on what we were talking about before first. So if you take a look here, we have our reactants and we have our products. And there is an energy difference. So you can think of this as the guy on the diving board and the products, he's in the water, and you can measure that energy difference, and that energy difference is delta G. So we talked about that already. But sometimes reactions have something called an activation energy. And that means that it needs a little something to get going. So you can think of something like gasoline or any fuel. Often those fuels, they could easily react with oxygen, but they don't. You need a little spark, you need a match just to get the whole thing going. And then once you have the match, you're making a fire or whatever. And chemical reactions are like this. And you can kind of think of it as, you've got these chemical bonds, they're kind of stable. But what you have to do is loosen them, right? You got to loosen them up a little bit first, and that takes energy. And that energy is called the activation energy. So you may see this as represented as different things. Sometimes I've seen activation energy like this. And there's other symbols that are used. Our textbook is using EA, which means energy of activation. So what is an enzyme doing? So an enzyme is finding a different way to do this whole thing. And so what you can see is here's the normal reaction without the enzyme. And with the enzyme, the energy barrier is just a little bit lower. And I'm going to talk about in a minute about how that is done. I've got a slide or two that show you how that's done. Before I do that, I'm just going to play this video for you. And this is a video from the textbook. It's a little bit old, but I like it because it uses the exact same images that the textbook uses. Plus it's kind of short, which is kind of nice and sweet. So I'll play that for you right now.
If exergonic reactions occur spontaneously, what keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction.
Okay, so he kind of repeated a lot of what I said, but I kind of liked how he did that and with the animation. So let's pick apart a few more of those details. Forgot about this slide. I meant to put this one a little earlier. Maybe I'll just come back to that in a second there. Hold on. Yeah, okay. So I'll come back to the energy of activation in a minute. There's a few other things I want to say about enzymes before we get more into that. One thing about enzymes is there's different types. I'm not asking you to know all the different types of enzymes, but you may recognize some of these terms. And most of them you can see end up with these last few letters, A's, A-S-E. And you can see this is giving some examples of the different classes of enzymes. And you may see these in your travels if you take more biology classes. Here's some exact examples of enzymes, and usually they're named, the name of the enzyme will list the substrate or the thing that it does, and then finish with A's. So you can see here's an example, hydrolase. So hydrolase is involved in hydrolysis reaction. So that's the thing that it does a hydrolysis reaction. DNAase, that's the substrate, it digests DNA. Proteinase, you can probably guess what that one is. It digests protein. A lyase, you may not know what that means because it's a specific type of chemistry, but you're probably familiar with oxidation reduction reactions, and there are oxidoreductases. And the list goes on and on. Transferases, they move something from one thing to another. Isomerase, they rearrange atoms so they make isomers. A ligase, this is something we're going to talk about with DNA synthesis, so we'll come back to exactly what that is a little bit later on in the semester. Here's some examples of some things that enzymes can do. They can break things down. We talked about sucrose. There's sucrose breaking things down. You've got your substrate. The substrate, by the way, is the molecule that is reacted on by the enzyme. It binds the enzyme at its active site, and we'll talk about that in a minute. And then you end up with your products. So notice this actually has two substrates. It has sucrose and it has water, and it has two products. So there's a whole bunch of variety on what enzymes can do with these kind of things. It can do the opposite. It can build things. I don't know what happened to that image. There's a big white blob on it. So you can see this has two substrates and it has one product. So huge variety of things that enzymes can do. Here's a little animation, and you can see there's two substrates going in, and you're going to see one product coming out. There it goes. Sometimes they transfer things, and you can see in this case here, you've got a molecule A, and you've got another molecule B and C, and the B gets transferred and it gets attached to A. I'll show you an example of this, like transferring a phosphate group. So if you take a look at this here, we have ADP, we have a phosphate on a substrate, and that phosphate gets transferred over and we make ATP. So that's a transfer reaction. So enzymes can be large, complex molecules. Some of them are very complex. This is one we're going to be talking about after the break that makes ATP. It's called ATP synthase. It has something like 15 or 18 subunits, and you can see it's actually quite complex. So we'll talk about that one a little bit later on. So we talked about the substrate. This is the molecule or molecules the enzyme acts on. And we also have the active site. So the active site is where the substrate binds. And so this active site is a custom chemical cleft. So that means that if the substrate is polar, the active site is usually polar, so that they can match each other complementary. Okay, and I'll show you an example of this. I have an active site here, right here. And if you take a look, this molecule here, RUBP, we're going to talk about this one when we get to photosynthesis. So notice this RUBP has this negatively charged phosphate here. And whereas over here on the enzyme, we have this positively charged polar hydrogen. We have another positive charge thing here going on. And that's kind of how an active site works. Like I said, you've got the size and complementary chemical properties on these things. These other slides here, we're just showing some other active sites. You can see there's lysozyme. Lysozyme cuts peptidoglycan in bacterial cell walls. Here is DNA polymerase. And if you look carefully, you can actually see two strands of DNA, that green one and this purple one here, or pink one, that are being produced by the enzyme. Okay, so this slide I thought was earlier, and this is the one where I said I was going to talk about how do they lower the barrier or energy of activation. So you can think about this as how do enzymes do what they do? How do enzymes do what they do? So they're sort of showing that in the three diagrams here. I was looking for a really good diagram. I couldn't find the most amazing one. But let me talk about what those three things are. So there's really three different things that are going on. So one is something that we call the proximity effect. So proximity effect. So what does that mean? So that means that you've got two substrates that want to react together, and they're not going to react together unless they're actually together. So it's kind of like a dating service, bringing two people together that normally wouldn't necessarily meet. So we can say that this enzymes bring the substrates together. So two is something called the orientation effect. So orientation effect means that if you bring things together, you can't just bring them together, you have to bring them together in the right orientation. So if you're going to attach two Lego pieces together, if you're going to attach a truck to its trailer, you have to bring it in the right direction, right? And so that's another important part of this. So we can say orient substrates so they react. And the last one is a little hard to explain without, it's a little easier in class when I have a prop, but we'll call this bond flexing. Flexing or stretching. And that's kind of shown here on this last diagram here. And so bonds, if you want to break them, sometimes you gotta massage them, you gotta bend them back and forth, and that's going to help them to break. So we can say that this loosens the bonds, loosens chemical bonds. And this is something I could go into in way too much detail on in terms of how exactly that's done. But like I said, you can think of it as the bonds kind of getting just loosened a little bit so that they're a little bit more amenable to actually reacting. I think I've got a picture on the next slide. I used to have a picture anyway. Let me see if I have that on the next slide showing that. Here it is. If you take a look, here's this is lysozyme. I mentioned that a couple of minutes ago. And if you take a look, we've got this this carbohydrate has kind of a chair structure to it. And what happens over here is it basically gets bent. The whole structure gets bent a little bit. But what does that do? It provides an opportunity for the enzyme and ultimately this water molecule that's going to get in there and do the reaction. So there's a lot of reactions like this, and biochemists study these things and usually have an idea of how they work after lots of study. And we're not going to get into that in any more detail for today. Like I said, this is just a enzyme basics lecture. So something else to know about enzymes is that they don't necessarily always work alone. A lot of enzymes have associated things called cofactors. So what is a cofactor? A cofactor means it is some sort of molecule or ion, and it is not part of the peptide. So it's not an amino acid or anything like that. And it's helping the enzyme carry out its function. So usually these are divided into different types of categories. So you can see we have inorganic ions, so minerals, so calcium and iron and copper and magnesium and manganese and mobillion, nickel. There's a whole bunch out there. Magnesium and calcium and iron and copper tend to be kind of the most common ones. And then there's others that are organic compounds that are usually derived from vitamins and non-vitamins. And you can see there's a list of a whole bunch there. So biotin, riboflavin, niacin, those kind of things are pretty typical. And so this is just, I want you to know this is a definition, really. What is a cofactor? Okay, so quick break here just to test ourselves. True or false? Enzymes change the amount of free energy required for a reaction. So the answer is false. What do enzymes change? Enzymes change the activation energy, not the overall energy of the reaction. So that is a different variable, and we're representing that as E little a. Number 2, true or false? A cofactor is made of amino acids. This is definitely false. And you saw in the previous slide, that was by definition, it was a non-peptide component of an enzyme. So it could be made out of all sorts of things other than amino acids, minerals, vitamins, those kind of things. So a few other things to say about enzymes. One thing about enzymes is they're pretty picky. They tend to have physiological situations where they're optimal. And you can see that's what really this is showing here. Is if you take a look at this top graph, you've got this, this little bar and showing that most human enzymes are most active at around 37 degrees. That's body temperature. So that kind of makes sense. If you're an organism that lives at a hotter temperature, then the enzymes are usually mostly active at those hotter temperatures, and so on. The bottom graph is showing that different enzymes might have different optimal pHs based on what part of the body they're found in, and so on. So kind of just a little footnote on this whole thing. A lot of enzymes are only active when they're found in the correct location. So this is showing the mitochondria and just saying that there are all sorts of enzymes there that are essential for respiration, and they don't really do anything unless they're in the mitochondria. So the last thing I want to talk about a little bit is introduce a concept that we're going to come back to when we talk about respiration, and that is regulating enzymes. And there's different ways to regulate enzymes. And one way to think about regulating enzymes is to turn them off. And we call this particular process inhibition. So there's that word, inhibition. There's different ways to inhibit enzymes. You can see they're showing two different types here. We've got competitive inhibition. And in competitive inhibition, we have an inhibitor here that is binding the active site and therefore blocking the substrate. Non-competitive inhibition, it's binding somewhere else on the enzyme, and still it's affecting the active site by usually changing the shape of the enzyme. So I have a little video that's gonna go over these processes, a couple other things to talk about, and then a couple of things we're gonna leave till after the break. So I'll play this video for you right now. It's another nice short one, and it explains this idea of enzyme inhibition. So.
During the normal enzyme catalytic cycle, the substrate encounters an enzyme with a specific active site to which it binds, forming an enzyme-substrate complex. The enzyme then facilitates the breakdown of the substrate to its products, which part from the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again. Competitive inhibition occurs when an enzyme encounters a blocker, which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur. Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as the non-competitive blocker is bound to the enzyme.
Okay, like I said, nice and short and sweet. And I'll show you some actual inhibitors of some enzymes. And actually, a lot of inhibitors are used as drugs. So this inhibitor here, AZT, this is a competitive inhibitor of an enzyme found in HIV, virus that causes AIDS. And it actually was the very first AIDS drug, a really breakthrough drug. And you can see that the drug there is very, very, very similar to thymidine. It's just different in that one little place over there where those nitrogens are found. So this binds the virus very tightly and does not bind human cells very tightly. And so it inhibits the viral replication. I've got a couple other examples here to show you. Here's another example of an inhibitor. This one is ASA. Some people may recognize what this is. This is actually aspirin. And so this binds enzymes that are involved in pain and inflammatory processes in the human body. And maybe one of the most widely used medications on the planet, in fact. Here's another one we talked about already, penicillin. This binds something called penicillin binding proteins in bacteria. And so if you take a look, here's my bacteria, and you're affecting the cell wall synthesis. And what happens, the penicillin gets in there and holes form in the cell walls. So quite an effective antibacterial drug. Works really, really well. So some inhibitors are bad, bad, bad. Here's one that is a nerve toxin. It's called sarin. This was actually used for a couple of terrorist attacks, one that was very notable in the subway of Tokyo a number of years ago, and you can look that one up. And what I want to do is just kind of finish off on one more concept that I'm going to get into with respiration. And just want to talk about how we can regulate enzymes both by inhibiting, but also by promoting activity. And this kind of inhibition, or this kind of regulation, is often called allosteric regulation. So take a look at this diagram. It's showing my enzyme, and the enzyme is, you can sort of think of it as like a hand. It's a flexible molecule, and your hand is sometimes open, sometimes it's closed. And if you're going to catch something, you want to have it open, want to have in the right orientation. And when your hand is closed, you're probably not going to be very good at catching something. And same thing with enzymes. They wiggle around these molecules. They're, like I said, they're relatively flexible. And so what we can do is we can actually stabilize one form or another. The inactive form, hands can get tied up, enzymes can get inhibited. And what that does is it shuts an enzyme off. We can also activate an enzyme. We can turn one on. And both of these things can be done. We're going to talk about how this is done in glycolysis when we get to that topic after the reading week. I'll show you one example here from the textbook. This is showing a pathway for the synthesis of another amino acid. So this amino acid is isoleucine, and we are using threonine to produce it. So notice this process, it takes 5 enzymes. So the way this works is that if you have enough isoleucine, you don't want to produce anymore. That takes energy. So if you have enough isoleucine, there's a little bit of feedback inhibition right here. And what happens is it goes and it inhibits this enzyme, this one here that's early on in the whole stage. So that's kind of how it works, right? You've got an end product works as an inhibitor for that enzyme, and it's allosteric. It's not binding the active site. Now you're probably wondering what happens if you have enough isoleucine. It gets used up by the cell, and this whole thing basically gets turned on. Anyway, let me take a look. So that's what I wanted to talk about today. Like I said, I want to come back to and kind of go into this this example of what happens in glycolysis, and we're going to talk about glycolysis after the break. And other than that, my goal was to finish a few minutes early today, and it looks like I've done that. So that is the end for today, and hopefully everyone has a great break, get caught up on everything, and come back after the break all refreshed and ready to learn some more biology.